Welcome back to the Genetics Lecture Series. Today we're going to be talking about sex chromosomes and some of the genetic conditions that arise from chromosomal imbalances and single gene variations. Before we begin, I want to briefly address the difficulty in talking about sex chromosomes and their related disorders without using gendered language some of the time. For the purposes of this podcast, when I refer to females or males, I am referring to the biological sex they were assigned at birth. For simplicity and to match common language in genetics and medicine, I have occasionally used the words maternal and paternal, which simply reflect inheritance from either an egg or a sperm. Okay, let's get started. X and Y are the sex chromosomes. An individual's sex is typically assigned by the external genitalia they are born with, which is determined by the presence or absence of the testis-determining gene SRY on the Y chromosome. Essentially, the absence of the SRY gene will result in female genitalia, and the presence of the SRY gene will result in male genitalia. While some autosomes do play a role in sex determination and differentiation, the sex chromosomes have the biggest impact. The X chromosome contains approximately 900 genes. By comparison, there are less than 100 Y chromosome genes. There are not many clinically relevant diseases associated with the Y chromosome. As such, we will be focusing on X-linked genes and their clinical impact, which affect all individuals. The XY chromosome complement suggests that gene expression from only one X chromosome is sufficient in males. The question is, do females express both X chromosomes or just one? The geneticist Dr. Mary Lyon proposed a hypothesis that those with two X chromosomes are in fact cellular mosaics with only one X chromosome expressed in each cell. Her hypothesis went on to be proven for the most part correct. Early in embryonic development, one X chromosome is usually chosen at random in all the somatic cells of females and is inactivated, a phenomenon we call X chromosome inactivation, which is also sometimes called lyonization. The asterisk here is that on the inactivated X chromosome, about 15% of genes continue to be expressed. And while X inactivation is usually random, structurally abnormal X chromosomes seem to be selected for inactivation more than not. Usually, the mosaic split of cells is about 50-50, with about half the cells expressing the maternally inherited X chromosome and the other half expressing the paternally inherited X chromosome. Even still, 90% of phenotypically normal females will have a distribution that extends from 25% maternal and 75% paternal, or vice versa. X chromosomes can impact human health from both the chromosomal level and from the gene level. Much like autosomes, sex chromosomes can undergo non-disjunction and missegregation resulting in aneuploidy. Two of the most well-known and studied aneuploidies of the sex chromosomes are 47XXY and 45XO. Individuals with Turner syndrome are monosomy, or in some cases partially monosomy, for the X chromosome. Turner syndrome is characterized by short stature, ovarian dysgenesis resulting in delayed puberty and infertility, and intellectual disability, along with increased risk for other medical conditions, including kidney dysfunction and cardiac anomalies. While Turner syndrome is diagnosed in between 1 in 2,000 and 1 in 5,000 births, less than 1% of zygotes that are monosomy for chromosome 45 result in a live-born infant. Patients with Turner syndrome have genitalia associated with the XX chromosome complement and are assigned female at birth. The mechanism for Turner syndrome is not well understood, especially since we just discussed that one X chromosome is inactivated in all cells anyway. I mentioned, however, that there are some genes on the inactivated X chromosome that are still expressed, which suggests that for some X-linked genes in individuals without a Y chromosome, the ideal gene dosage comes from two X chromosomes, not one. Klinefelter syndrome is found in up to 1 in 650 births of those who appear phenotypically male. Individuals with Klinefelter syndrome are typically tall and thin, but it is not until puberty where signs of hypogonadism begin to become apparent that they present for clinical assessment. Testes remain small, and sexual sex characteristic development is stunted, sometimes resulting in a diagnosis of delayed puberty. Just like single-gene autosome disorders, X-linked disorders are classified as either dominant or recessive, but the way we use these terms is a bit different. Whether an X-linked condition is considered dominant or recessive depends on the phenotype expressed by a female who is heterozygous for the gene. If the phenotype of a particular mutation is not observed in a heterozygote, it is considered a recessive condition. If a phenotype is consistently observed in a heterozygote, it is referred to as a dominant condition. This might seem like an obvious classification system, but because of the X chromosome mosaicism in females due to X chromosome inactivation, there are many pathogenic alleles that may or may not present with the phenotype in heterozygotes, depending on the proportion of somatic cells that express the pathogenic gene. To illustrate some of the differences between the inheritance patterns observed in autosomes and sex chromosomes, let's take a look at a case. Jeremy is 6 years old. His parents initially became concerned when Jeremy stopped climbing stairs, and while he used to run all the time, he would tire quickly and mostly just walked now. Jeremy's family doctor reviewed developmental milestones with his parents and learned that while Jeremy met all of his milestones at an appropriate age, he seemed to have regressed in some of the gross motor milestones. On exam, he thought he could appreciate some proximal weakness and maybe some tightness in Jeremy's heels, and he noticed that Jeremy was unable to climb up onto the examining table without assistance. He referred Jeremy to a neuromuscular clinic at the Children's Hospital. The specialist group met with Jeremy and his parents, who revealed that they and Jeremy's 2 older sisters were healthy. When examining Jeremy, they observed the same physicality that the family doctor did, but they also noticed that his calves were enlarged, and he rose from the ground in a series of maneuvers that are referred to as a Gower sign. They ordered a creatinine kinase, which was quite elevated, and they clinically suspected Duchenne muscular dystrophy, a condition that is characterized by muscle degeneration that begins to become apparent around the age of 5. Before we continue, let's just think about how we inherit our sex chromosomes so that this all makes sense when we think about inheritance. An egg can only provide an X chromosome to a zygote, which means it is the sex chromosome provided by the sperm that determines biological sex. If the sperm provides an X chromosome, the zygote will be phenotypically female, and if the sperm provides a Y chromosome, the zygote will be phenotypically male. Genetic testing confirmed that Jeremy does indeed have the pathogenic Duchenne muscular dystrophy allele on his X chromosome. Since Jeremy could only inherit his Y chromosome from his father, he must have inherited the X chromosome from his mother. Indeed, his mother carries the pathogenic allele for Duchenne muscular dystrophy, but has the wild-type allele on her other X chromosome, so she does not have the phenotype and is a carrier. Additional testing shows that Jeremy's sister and his maternal grandmother are also carriers for the condition. Often, pathogenic alleles displaying X-linked recessive inheritance are passed through multiple generations through female carriers, which is what has happened in Jeremy's case. It is rare for a female to have an X-linked recessive condition because that would require her to receive an effective allele from both her mother and her father. This is extremely unlikely, though, due to how rare most of these conditions are. For this reason, males are far more likely than females to be diagnosed with a recessive X-linked disorder. X-linked dominant conditions are far less frequent than X-linked recessive conditions, and they are a bit more nuanced. As a reminder, we have defined an X-linked dominant condition as one where a phenotype is expressed in a heterozygous female. A fully penetrant X-linked dominant allele will affect all individuals who have the allele, regardless of their sex chromosome complement. Affected males will pass on their affected X chromosome to 100% of their female progeny, but none of their male progeny, so X-linked dominant conditions are never passed directly from male to male, and a male will always pass on the condition to a female child. An affected female can pass on her pathogenic X chromosome to both male and female progeny, with each child having a 50% chance of inheriting the disorder like we see in autosomal dominant conditions. Usually, the phenotype is milder in females compared to males either because of X inactivation or because they are heterozygotes. In fact, there are some X-linked dominant disorders that are lethal to males before birth. Rett syndrome is an X-linked dominant neurologic disorder that is usually lethal in hemizygous males, and as such occurs nearly exclusively in females. We have highlighted the important role of sex chromosomes both in their function of sex determination and differentiation, and their role in genetic conditions. Understanding the nuances of X chromosome inactivation and how X-linked disorders present informs clinical practices and genetic counseling, ultimately aiding in the management and treatment of these conditions.